Hello! Hello and welcome to the miniPCR webinar. Thank you for joining us. My name is Bruce. I am the Director of Curriculum at miniPCR Bio. And again, we're just really so glad that so many people have joined us here today. Like so many of you, we are socially isolating. I am here in my house in Rhode Island. As you all are too, I'm sure, at your houses all over the country. So what we're hoping to do today, we are hoping to give you a resource that teachers you can use in your classroom. So this is intended for students to be able to watch this and sort of have some of the experience of doing the miniPCR PTC Taster Lab. As part of that, in the links below, you can find a worksheet that we've linked to that students can do. Students could also follow along on the regular PTC lab guide. Also for teachers, we know that some teachers were interested in this because you may be interested in implementing it in your classroom in the future. If that is the case, you'll see how I would teach the lab. You can also ask questions as we go in the chat, which is just off to the side. I have a couple colleagues there that will answer your questions. All right, so let's dive into it. Okay. So the PTC Taster Lab, from genotype to phenotype. So just to be clear, when you get a lab, this is what you get. It's a bunch of tubes, and we'll go through every one of these tubes as we go. But today we're going to do this classic lab from beginning to end. And I like to just frame this lab with this, real sort of basic question, right? A lot of times in my human physiology class, I'd have a student come in, ready to blow my mind, right? Mr. Bryan, have you ever thought, like, is your blue my red and my red your blue? And it just gets to this question about perception, right? I've grown up, people have shown me an apple. You've always said this apple is red, and so that's what I call red. But how do I know that in my mind's eye, I'm seeing one color and you might be seeing that apple on the bottom and someone else might be seeing that apple in the middle? How do we crack open how we perceive the world and whether we perceive the world the same way? Something like this, it's very difficult to do, right? I really don't know how you can crack open all aspects of perception. For something as dramatic as this, it's almost certainly we probably see the same red. Right? We are all humans. We share an evolutionary lineage. We go through the same developmental pathways. I'd be really shocked if something as dramatic as my red is your blue. However, we do know there are differences in perception. And when those differences become apparent, it's pretty exciting and fun. And one of the most classic examples of this is, I think, from 2015, this dress that is like so obviously white and gold. And if you say anything different, I really think you are insane. However, my wife claims the dress is blue and black. And she thinks I'm insane. What are we going to do? Breaking something like this down and understanding how we get to white and gold versus blue and black, there's probably so many layers on what's going on and in our brains as to how our perceptual systems work to get to those differences. I've seen some suggestions on how it might work, but really nuts and bolts, we don't know. And there are some interesting things that we do understand, though. One of the real classic ones is cilantro. Right? If I have a taco, I love a lot of cilantro on it. One of my good friends will not touch cilantro. It tastes like soap to him. That sounds crazy to me, but that's the way it works. And that's a system where people actually understand some of the genetic underlying mechanisms that are going on. However, probably the most classic case of this is what we're talking about today, is PTC tasting. So PTC stands for phenylthiocarbonide, which is a word you'll never have to remember. It's a chemical normally found in foods. I actually tried to look up for this what it's normally used for. I couldn't find anything other than tasting it. The basic idea is that if you put this chemical in your mouth, and usually you get on a little piece of paper, you get two responses. Either like, ew, that is gross, I don't want to be tasting that. Or I have no idea why everyone is grossed out. It just tastes like paper. Little side fun note, the way PTC, that this response was discovered was in a chemistry lab. Someone was working with PTC, and they spilled it, and the powder went up in the air as a fine dust, and the person next to him was like, oh my gosh, that tastes so bitter, and the chemist looked over and he said, what are you talking about? I don't taste anything. And so like any good chemists, they repeated the experiment and kept putting it in their mouths until they'd sort of worked some of this out. So we're doing a lab. Let's do it. So this is PTC paper. Now, some folks ask, can we replicate this at home? PTC, one of the things that's so special about it is that it works in such a clear way, and that's why we use it. So I don't know of any other chemical other than other chemicals that you get on little papers like this that work like this. So replicating PTC at home would be very difficult to do in this lab. However, the paper is very cheap. So you can buy a tube of this paper for like $5 on Amazon. You can get it from Carolina or from any biological supply company. It comes with 100 pieces of paper in it. So all you do, you put it on your tongue. It's important to use control paper too, because you want to make sure you just, it's not that you just don't like the taste of paper, or you want to make sure you're really tasting something. So I'm going to do it. I'll start with my control paper. It tastes like paper. And now, the PTC. Yeah, it doesn't taste good, that's for sure. Some people really react negatively. I wouldn't put that on my breakfast cereal, but it's gross. It's not the worst thing in the world. It's definitely not good though. But because I can say that, because I can tell the difference between the two papers, that definitely means that I am a taster. So I'm going to record my response here as this. That is me. That is my tasting face. And I'm going to give myself a big T because I can taste it. And we'll get into a little more detail on what that means in a little bit. Now, normally I would do this with my class. However, I don't have a class right now because I'm not a teacher anymore, but I'm also stuck at home. And so the only people I have to test this on is my family. And so they didn't have any choice. All right, so I gave it to my older daughter. This is her official response. That is a tasting face. She really does not like it at all. I gave it to my younger daughter. That is a tasting face. She doesn't like it either. Her response is a little bit more like mine. She's not crazy about it, but it wasn't that bad. And finally, we gave it to Mom, and that is another tasting face. So this family is 4 tasters, though, and we'll get into this a little bit more later on, perhaps, but it is important to note that in our tasting, we have differential response. So my older daughter, actually, she tried it as a freshman in high school, and she actually begged me to not have to taste it again because she remembered how bad it tasted. But like any good dad, I made her. Where, again, her mother felt similar. Where I and my younger daughter, it's bad, but not that bad. All right. So let's talk about just what happened there real quickly. So to taste something, you need the chemical to bind to taste receptors in taste cells. So this is your tongue. If you look closely, you'll see there's lots of little white sort of dots on it. Those are called fungiform papillae. And those fungiform papillae are where the taste buds live. They're not taste buds themselves, that's where the taste buds live. In each fungiform papillae, there is about 3 to 5 taste buds is what I understand. So if we zoom in on here, this is what one taste bud would look like. And each one of these taste buds has a few dozen taste cells in it. So as you take the PTC paper, that chemical dissolves in your saliva, it washes across your tongue. As it comes across one of these fungiform papillae, it will go into the pore, it will touch some of those hairs, and if it touches the correct receptor, it will send a response that will then go off to your brain. Just a quick note, there are other types of papillae. there's more taste receptors along the side of your tongue, along the back of your tongue, and also along the side, some in the sides and the back of your mouth. So in this lab, what's so fun about this lab is that we can get this full picture of genotype to phenotype. I've already taken my phenotype, right? I'm a taster. But now for this lab, we actually know the genotypes. And so we're going to be able to figure out exactly what my genotype is. And because the mechanisms are so well worked out, we're going to be able to go along and follow this through transcription. We're going to talk about what the differences in the proteins are. And from there, we're going to talk about how does those difference in the protein, how does that signal actually make it to your brain? And this is what's so great about this lab, is because it really does connect all the way from genetics to perception in a really, really great way. Quick overview. Normally, this would be a 2-day lab. That's what we normally consider it. You can do it in 2 45-minute periods. If you were to do it in one period, you'd probably need about 2.5 hours, something like that. We are not going to take that long today. Just so you know, I'm going to be doing a little bit of the cooking show version of this, where I'll be demoing it and then hitting a little fast forward, especially on the PCR stage. That takes about an hour. We're not going to wait an hour for the PCR to run. Okay. So just jumping back in, the receptor that we're talking about, we were talking about washing over the tongue there and binding to the receptor. So the receptor, the protein that we're talking about, is the TAS2R38 taste receptor. It's a bitter taste receptor. It's part of the T2R family of taste receptors, and that name TAS2R38 means there is a TAS2R1 through 37 as well. So there's a whole bunch of these taste receptors that are very similar. They just bind to slightly different molecules on the surface of the protein. It is a Gprotein coupled receptor, and if you are sort of like an intro bio, you probably don't need to worry about that too much. If you are an AP or a college bio student, you'll probably be familiar with that term. And that is how all sweet and bitter taste receptors work. They're Gprotein coupled receptors, and structurally they're all very similar. Salty and sour taste receptors are ion channels, and I'm not sure how umami works, but that's for another day. So let's take a little bit closer look at how this works. And again, we're going to go over this super quickly. And if you're an intro student and this looks like total chaos, you've never seen anything like this before, just hang on. We'll be back to you in just a second. But it is important to realize that when you have a sensation, there's sort of a couple steps in this process. You have the chemical binding, you have a transduction pathway, and then you have a response. So let's just break that down real quick. So this here, that little oval represents our ligand or our PTC molecule. If it binds to the receptor, that's stage number 1. If it binds, it sets off this transduction pathway. Gasteucin, a secondary messenger, is activated. That's your Gprotein, and it has a signaling cascade that goes off through the cell. And then finally, there's a response to the cell. And the response in this case is going to be the releasing of neurotransmitters and the starting of a nerve signal to the brain to tell you that you just tasted something bitter. I just bring that up because Gprotein coupled receptors are such an important biological concept. They are used all over the body for all different types of things. Hormone receptors are a common example of them, but here they are in taste, they're actually in vision. They do all sorts of things, and the signaling cascade is really well understood, which is why we can use it as this example for PTC. And just the little side note there, it's the target of something like 1/3 to 1/2 of all marketed drugs. So incredibly important physiological types of protein. Okay, so to test ourselves, we need to get DNA. So I'm going to do that here. We're just going to do a quick DNA extraction. So in this tube here, I have 50 microliters of our extraction buffer. And our extraction is really, really simple. I'm just going to take a toothpick, and I'm just going to rub the inside of my cheek 3 to 5 times max. There we go. And then I just twirl it and go away. And then we're just going to do a real easy DNA extraction. Oh, I have a tube in there. Whoops. I'm going to use right here. I'm going to put it in my miniPCR machine. And I'm just going to use this as a heat block. So we're just going to heat it up to 95 degrees real quick. And I need to get my miniPCR program going to do that. So here we go. So we're going to, I have a program here called Make It Hot. That'll do 95 degrees for 10 minutes. And I'm going to do run. It's going to take about a minute to load, and then I'll show the temperatures there. So just a real quick note on this, we do a real sort of quick and dirty DNA extraction. If you've done DNA extractions in labs before, you may do like a full ethanol precipitation, which involves lots of spinning and pipetting. Ours, we just use an alkaline lysis buffer, so it's got a pH that helps sort of open up the cells, and then we cook it, and that just pops open the cells. That means in our future steps of the procedure, we're going to have all sorts of stuff in there. We're going to have mitochondria. We're going to have endoplasmic reticulum. It doesn't matter. We just want that little DNA that we're going to get out as well. If you do a much more complex DNA extraction, you're probably going to get much more pure DNA at the end. But for what we're going to do, it doesn't matter. So we can do this in 10 minutes. And you're much less likely to have an error because there's only one pipetting step and a 10-minute incubation. Okay, so we're getting out our DNA. Why do we want our DNA? Well, we know the genotypes for this TAS2R38 gene. We know the different alleles. And for this gene, there are two main alleles. Just to be really clear, there's more than two alleles, but the vast majority of people fall under these two main alleles. And these alleles are differentiated by 3 SNPs. This SNP there, that you pronounce it, you say SNP, and that stands for single nucleotide polymorphism. So poly, right here, poly means many. Morphism, morph is a form, so we have more than one form of these alleles, and they're different because of single nucleotide changes. So when I go down what that means is I go down the genes, it's not going to be a major insertion, it's not going to be a deletion, it's not going to be totally different. As I go along and I compare the A's and T's and C's and G's, a SNP means that they're all the same until you just get to one, and one is an A and the other one's a G, or one's a C and the other one's a T, something like that. So between these two alleles, there's 3 times when there's a single difference between them. So let's look at them. So as I go along, the first one I find is at base pair 145. Oh, I should step back real quick. This gene is 1,002 base pairs long. There's no introns. It's an incredibly simple genetic structure. So 1,002 base pairs. So at base pair 145, on the taster version of the allele, there's a C. On the non-taster allele, there's a G. And that's on the, I'm looking at the coding strand there. I go along, again, nothing of interest until I get to base pair 785. At base pair 785, on the coding strand of the taster allele, there's a C. On the coding strand of the non-taster allele, there's a T. And again, all the same until you get to base pair 886. On the coding strand of the taster allele, there's a G. On the non-coding strand, on the coding strand of the non-taster allele, there's an A. So those are the 3 consequential differences between these two alleles. Let's see what those actually do to the protein. So when we look at this, we see the DNA here. And the important thing is that DNA makes a messenger RNA to make the protein. So we can look at these messenger RNAs. And these messenger RNAs are copied off the bottom strand, which is the template strand there. So they're going to look just like the coding strand, except when you get to a place like right here, that T on the coding strand is a U in the mRNA. But otherwise, they're the same. And this is just to say that those changes, those SNPs in the mRNA get transferred right over, I'm sorry, those SNPs in the DNA get transferred right over to the mRNA. So let's look at what this means for a protein. So as we read the mRNA, we make an amino acid chain, and that is our protein. So in this, all these little dots here represent the amino acids. And then we get here, the place that we're interested in, are arginine, Q. Oh, I should have double checked that beforehand. I believe it's glutamine. And then P stands for proline. When we look at the taster allele, when we translate this mRNA into protein, we have all these amino acids, and then we get arginine, glutamine, I think, and then alanine. So proline and alanine. We go along up here, serine, cysteine, alanine. On the bottom, serine, cysteine, valine. Again, in the taster allele, alanine, alanine, valine, and in the non-taster allele, alanine, alanine, isoleucine. So those three SNPs result in 3 amino acid changes in the protein. Now, sometimes, because this is proline, alanine, valine, this taster allele is sometimes called the PAV allele, where the non-taster allele is referred to as the AVI allele. I'm going to try to not to do that just because it gets confusing when you're using taster, non-taster, PAV, AVI. There's too many variables. But just if you do see the PAV, that's what PAV refers to. It's the taster allele. It refers to those 3 amino acids. And AVI refers to the non-taster allele. It refers to those 3 amino acids. So up here is the protein as it looks sort of 3-dimensionally when I showed you that before, inserted in the membrane. Here we've taken it and we've sort of opened it up so we can see the whole amino acid chain. And the important thing here is that I've just pointed out where those PAV and AVI changes are. So PAV in the taster allele and the AVI, the non-taster allele, looks exactly the same, just at those 1, 2, 3 places we've made small changes. So we just show this to you because actually people aren't 100% sure why non-tasters can't taste. As we showed in that Gprotein coupled receptor slides, there's sort of 2 things this protein does. It receives the ligand, it binds to the PTC chemical, and then it sends the signal inside the cell. So that's sort of two ways that this protein could break. We just show you this because if you note where the changes are in the cell relative to the cell membrane, right? If this is the cell membrane, the outside of the cell and the inside of the cell, you can start to make a prediction about which one of those 2 things are different. Now, again, we don't actually know for sure, but we can make a prediction. Okay, so we do this, one of the reasons that people love this lab, because it teaches classical Mendelian genetics and Mendelian inheritance. So the TAS2R38 gene is located on chromosome number 7. Chromosome number 7 has about 1,000 genes on it, and TASR238 is just one of them. It's a pretty small gene, small simple gene, sort of off to one side here. And there's 2 forms of it, right? So we said the big T we're going to call it now is the taster allele, and the little t we're going to call it the non-taster allele. So now just to be really clear about the different genotypes you could get, because everybody has 2 chromosome number 7s, everybody's going to have 2 copies of this gene. You're going to have 2 alleles, right? If you have if you get a chromosome from number 7 from your mom with a big T and you get one from your dad with a big T, you're going to be a taster. You're going to be a homozygous taster. If you get one from your mom and your dad both with little t's, you're going to be a homozygous non-taster. And then the third option is you could get one chromosome with a big T and one chromosome with a little t, and you would then be a heterozygous taster. So we call this dominant because both the homozygous dominant taster allele and the heterozygote are both able to taste the PTC chemical, and the little t, the non-taster down here, is not able to taste it. So to find that gene and to figure out what you are, we have to do a process called PCR, the polymerase chain reaction. PCR, as I said, that chromosome is 1,000 genes long. We're just looking for one. That's one chromosome out of all the chromosomes you have. And so we have this really complex DNA sample that when I just scrape my cheek, I put it in that tube. Now, I just ate a banana with peanut butter on it, so I guarantee there's peanut butter DNA, or peanut DNA. There's banana DNA in there. There's my DNA in there. There's bacterial DNA in there because there's bacteria that's living in my mouth. So we have in that tube that's still heating in the machine here, we have a really complex DNA sample. We need to key in on that one region of interest, and we need to find the DNA that we're looking for, and then we want to make a whole bunch of copies of that, because that's really all we care about, is that one PTC gene. So that's the job we're going to do with the polymerase chain reaction. To set up a reaction, we take one of those little tubes that we had before, and in it we put the DNA to be amplified. That's the DNA that I took out of my mouth and is boiling right now. We need primers. So primers are really special sequences of DNA that you design for every experiment, and they come as part of the lab. They come as one of these little tubes here. I'll take it out in just one second. Those primers mark the beginning and the end of the gene that you're interested in. So the primers are what find the DNA that you want to make copies of. The next things that we need to put in our DNA polymerase. So if we're going to make copies of DNA, we need the enzyme that makes copies. The polymerase that we use is called a Taq polymerase. It comes from a thermophilic bacteria. That we will understand why that's important in a second. But basically, this is an enzyme that copies DNA, just like the polymerases in your cells that are able to copy DNA. But this one is able to get heated up to boiling, and it's not going to denature like most proteins would. Next, we're going to put in a whole bunch of dNTPs. dNTPs are the A's, the T's, the C's, and G's, all the building blocks that you need to make new DNA. If you're going to make DNA, you need the building blocks to do it. And finally, we're going to put in some buffer and to maintain pH, and in that buffer is also some magnesium chloride, which is a cofactor for the Taq polymerase, but we don't need to worry about that. When we actually do this, there's just 3 ingredients to put in. There is the template DNA, which we just made. I don't know if you can hear the dinging. I can hear it in my ear, but that's because the program just finished. So my DNA is ready. We're going to put in the primers. These 2 things are unique for every PCR reaction. And then the final 3 things here, these are basically the same. So we just give you what's called a master mix. It looks green. And that's how you set it up. So for this one, we are going to use 3 microliters of DNA, 12.5 microliters of primer, and 12.5 of PCR master mix. So I'm going to do it. There you go. I have my little lab set up here. I probably should have done this before, but I'm going to go ahead and glove up. All right. So this is all ready here. So this has my, this tube right here has my DNA in it. Oh, just quick side note. This is this little rack I use, a little lab tip. This is an old pipette tip rack. That's just a great way to get tube racks for PCR tubes, is use your old pipette tip racks. All right, so in there, I'm going to put 3 microliters of my DNA. There you go. So 3. And now I'm done with that DNA. I'm not going to use it anymore. I'm going to put it all the way in the bottom of the tube and get rid of my tip. Then calls for 12.5 of master mix. This is the green master mix. And then finally, it asks for 12.5 of the PTC Lab primers. And then I just close it. And to mix, I use this technique. You just give it a flick, and then you give it a shake, and it's all back down at the bottom. And we put it in the machine. If you're doing this lab in a classroom, each student would be doing 1 tube, so each, one tube per student. Again, I've done other ones previously that I'll show you the results for, but right now we are just showing you one. All right, so next so next I need to program my miniPCR machine. So this is the program. This is the software for this machine. It runs off of a, I didn't put it back up. It runs off of a smartphone, a Mac PC, basically any device, Android, running off a Mac right now. And so one of the nice things about these machines is that you can program them yourselves and that you can see it running right in front of you. So I'm going to just show you the program. We're going to do a new program. It's a PCR, PTC. We'll give it an exclamation point. All right, and then we said that the program was 94 degrees for 120 seconds, 94 degrees for 10 seconds. We're going to change the annealing temperature to 58, and that's going to be for 15 seconds. The extension is still 72, but we're going to change that to 40 seconds. And it calls for a really short final extension. The final extension usually isn't very important, so we're doing a real short one. And we're going to go for 30 cycles. So that's it. I'm going to hit save and run. When I do that, the lights on my machine should blink. I don't know if they did or not. It should blink 3 times. Yep, it started up. And so now my machine is running. I don't know why the program's flickering a little bit. It's not on my screen. That has to do with the YouTube interface. Okay, so we're going to let that go, and I'm going to come back to it in a second because right now I'm just going to explain what we just did. Okay, so this is the program. This is what we just entered in, but why did we do that? Okay, so PCR is a 3-step process. Step number 1, we said we heat it up to 94 degrees. When you heat DNA up to 94 degrees, it is going to denature. Okay, so this DNA is going to come apart into 2. Now just remember, right now this DNA is a really complex sample and those strands are really, really long. They're probably millions of base pairs long. So we're just separating it up, opening it into 2. Now the next thing we do, for this one we set it to 58 degrees, that's the annealing temperature. When we do that, things start to come back together because we're cooling it down. But in here, we've put in these primers. And these primers, I said, are what find the beginning and the end of the sequence. So the primer binds to the beginning and the end of the sequence because we've designed them to. These primers are 5 base pairs long, normally, or 5 nucleotides long. Normally, they're about 20 plus or minus a little bit. So we design special primers for every lab that are complementary to the beginning and the end of our DNA sequence. Then when we heat it up to 72 again, we have the Taq polymerase. And the Taq polymerase is this molecule here. And that is an enzyme that's going to find ends of DNA like our primer, and it's going to copy them. And it's going to do that by taking the DNTPs that you see out there. It's going to take all the DNTPs in solution, and it's going to lay them down and make a new strand. So we just went from one strand of DNA to two. And then we're just going to do that over and over again. We're going to heat these two strands down at the bottom up to 94 degrees. They're going to melt. Then we're going to cool it down. The primer is going to meet. And then we are going to add the tac's going to bind when we heat it back up again, and it's going to extend. That means that we go through exponential growth of our DNA. People have been seeing like a lot of exponential growth curves lately, so hopefully you get the idea, right? That it starts out kind of slow, and then you end up with a lot of DNA. So after about 30 cycles, which is what we're doing here, you should have roughly 1 billion copies of your DNA for every copy you started with. And again, that's 1 billion copies of only the very small sequence that we're interested in. In this case, in this lab, we're copying a 250 base pair sequence, just 250 base pairs of that gene. And we'll talk about why it's that little region in just a second. So in about an hour, we're going from, from every one, we're going to 1 billion copies of that sequence. Thermal cyclers have changed a lot over the years. When PCR was invented, it was actually just water baths, and you moved the tubes along by hand. A lot of hours were spent by graduate students doing that. The second generation machines, if you see ones today, they still sort of a lot look similar to this. That's one of the key differences for the miniPCR. We like to say it's not a black box. Really, it's a simple idea, right? We're heating and cooling to very specific temperatures. There's that metal, if you can see that right there, that's the heat block. It heats it up. When it's time to cool, in the middle here, that black part in the middle, those are fans. That's how it cools down. And then there's a circuit board on the bottom to retain the program. So let's check in on what's going on in the PCR. All right, so we're one cycle in. So right now it's telling us that it's doing the extension right now. It's heading up to 72 degrees. So it's going to be at 72 degrees for 15 seconds, and we'll just wait with it for 1 second. It also gives you a graph. Right now that graph is really boring because we're only on cycle 1 or 2 here. So we're at just 2 DNA copies to start. This will start looking like an exponential curve. So if you ever use these in class, it really just sort of opens up. We really just want to crack open this process so students can really wrap your head around what's going on. All right, and I just want to give it 1 more second. Here it goes. So then denaturation. So now it's heating up to the 94, and so that's when the DNA is actually denaturing and going from 1 copy to 2. All right, so that's just a little preview of the program. Let's jump back here. All right, so remember, we're genotyping my family. So dad, that's me, mom, and 2 children. So what do we know about the genotypes already? Well, we said that we're all tasters, and we said tasting is dominant. So we know that we all have that big T taster allele. What we don't know right now is what the other allele is. We don't know if we're heterozygotes or if we're homozygotes. If you've done this in class before, just with the PTC part, this is where you have to stop. So now we can take it a step further and match that DNA really to the actual phenotype that we have. So we're going to do that with restriction enzymes. You may say, why don't we just sequence it? Today, it is the era of sequencing. We could do that. You probably can't do that in a high school very easily. Sequencing has gotten very cheap and easy, but it's not quite that cheap and easy yet. So we're going to use a technique called restriction digest. It's a classic genotyping technique that's been used for decades. I used it a ton when I was in graduate school, and people still use it today. Restriction enzymes are enzymes. They come from bacteria. They're part of the bacteria immune response. They are able to recognize very specific sequences of DNA, and when they find that very specific sequence of DNA, they cut it just like a little pair of scissors. So this is a very common enzyme, EcoR1. Oh, 1 second, I just lost myself here. Okay, EcoR1. EcoR1 recognizes the sequence GAATTC. If EcoR1 is in a mixture with DNA and that sequence is there, EcoR1 is going to cut it right along that line. The one we're actually using today is an enzyme called FNU4H1 over here. FNU4H1 recognizes this sequence. GC, N means that it can be anything. So that N is not important. And then again, a GC. So GC, any old nucleotide, GC. And if it finds that, it cuts it just like that. So let's look at an example. Here's a DNA sequence. We go along, we're looking for GAATTC. I don't see it. All right. Now let's look for GC, any old nucleotide GC. So as we go along, we have GC, any old nucleotide GC. We found our recognition sequence right there, which means that if we put in the FNU4H1, it's going to cut it. And it's take that one piece of DNA and it's going to cut it into 2 pieces. So this is why this is important. If we look at SNP 785, base pair 785 in that sequence, this GC, any old nucleotide GC, we can find it right there in the taster allele. Which means that if we take that taster allele and we mix it with FNU4H1, it's going to cut the DNA into 2 pieces. Now let's look at the non-taster allele. This is where the restriction site was. You see that that C is now a T. So that FNU4H1 won't recognize that sequence. We mix the in with it and nothing's going to happen. So it just doesn't cut the DNA. So we can use that restriction fragment length polymorphism as a diagnostic. And that's what that RFLP stands for. Restriction fragment. We've made fragments, and it's another form of polymorphism. So it's another place where these 2 things are different. Now, just to be really clear about this, there's nothing special about FNU4H1. It just happens to, the recognition sequence just happens to have SNP 785. They happen to overlap. FNU4H1 would probably cut this gene in other places far down the way. It cuts your genome all over the place. But it just happens that it cuts in this spot. We're lucky that it does because we can use this to genotype. We're not that lucky. There are hundreds of restriction enzymes, so quite often when you find polymorphisms, quite often but not always, there is a restriction enzyme that will that the polymorphism will be part of the recognition sequence. So here's the thing, you may be thinking, all right, so if we can just cut the DNA, why did we have to make all those copies of it? What was the point of that? Well, if we think about this, our gene is 1,000 base pairs long, right? But the human genome is 3.2 billion nucleotides long. So imagine if you were to write out your entire human genome, that complex sequence that we had. We're going to write that all out on paper, and every piece of paper is going to have 500 nucleotides on it. Well, if you were to do that for the entire human genome, you would need a stack of paper 1/3 of a mile tall. Okay, so that's taller than One World Trade Center, the new Freedom Tower in New York. It's taller than that. That's how much paper, and that again, that's not end to end, that's like reams of paper stacked up. That's how much paper you need to write out your genome. We're looking for 1 nucleotide difference in a gene that is 2 pages long. So if we were to just mix FNU4H1 in with that pile, it's going to turn it to just, it's going to be strips of paper all over the place. And we're looking for 1 of those strips of paper. So what we do is we use PCR as a photocopier. We find that single page that we're interested in and we copy it. And then we do the trick of taking both papers back and put them through the copier again. So you go 1 to 2. And then you take all 4 and put them back through 4 to 8, and eventually you get 1 billion. When you get a billion pieces of paper, a billion piece of paper is a stack of paper 63 miles high. Okay, so we went from a third of a mile stack of paper, that's a lot, looking for one nucleotide, but now we have 63 miles of stacked paper, and every single page has the nucleotide that we're interested in. That's the power of PCR, and that's why we use it. So a reminder where we are. We've done a DNA extraction. We've run the PCR. We're about to do a restrict and digest. The DNA extraction, just a quick note on timing here. The DNA extraction that we do, because it's a little crude, you want to do that relatively quickly. You want to do the PCR relatively quickly after that within the same period. You always want to do those in the same period. That PCR can then run, and it takes about an hour, but PCR is stable, so you can like run and you can go home and come back 3 days later and the tubes will be sitting there and they'll just be great. So that's your stopping point, is right after the PCR. Then we're going to run the restriction digest and run the gel. Just so you know, this is a little bit tight for a 45-minute period. You can do it. It's a little more comfortable. I used to have 52-minute periods and we could do it there fine. 45 gets a little tight. If you have hour periods, something like that, or if you have long blocks, it fits really easily. If you want to stop after the restriction digest, that is an all right place to stop. Just know that you put the things in the freezer. Okay, a little off topic. Here we go. Restriction digest. So we're going to pretend my PCR is done. Yeah, open this down. We're going to pretend my PCR is done. So we're going to take it out. And in a new tube, we're going to take 14 microliters of our restriction of our PCR. Okay. So this is now set to 14. And the PCR now is really all just those taster and non-taster alleles. That's really basically all that's in this tube. The original DNA sample that we put in there is still there, but there's so little of it compared that it doesn't matter. And so we're just splitting this in half. Okay, so this is the one we're going to restrict. And then we are going to add 1 microliter of our FNU4H1. And so I just want to know I'm using a different pipette right now. I'm using the 10 microliter pipette because we're just using 1 microliter, and that's a very, very small volume. The 20 microliter pipettes usually don't go down to 1 microliter. We usually suggest that the teacher does this step because, well, 2 reasons. Restriction enzymes are fairly expensive, and so you don't want to mess this stage up. 1 microliter is very small. So no offense to students out there, but your pipetting skills are usually, well, they're still growing, we could say. And so we don't want to mess it up that way. And then finally, having a 10 microliter pipette, a lot of times just you might have 1 in your classroom instead of where you probably have a larger set of the 20 microliter pipettes. So I'm going to get my enzyme here. The enzyme, so there's only 50 microliters in this tube. The enzyme is also something you want to keep cold until you use it. Everything else is pretty heat stable. The restriction enzymes are pretty wimpy. If they get heated up too much, they don't like it. With all this stuff, but this one again, especially because there's so little, you really want to make sure that you spin this down in a centrifuge before you use it. If you don't have a centrifuge, that's okay. I don't here at home. I just do the human centrifuge flicking, but like, you know, really make sure you get it down there. Don't be afraid of a little tennis elbow or Tommy John surgery after you're done. You really want to make sure it all gets down there. All right, so I'm going to take out my 1 microliter. There we go. And I'm going to add it to here. Sure that the pipette's down in the liquid when you're pipetting. There we go. And again, I want to do my mix, flick, and again, my human centrifuge to bring the tubes back down. Okay. I'm now going to put it in the PCR machine. Just one thing, when you do this, make sure your PCR machine isn't hot anymore. I can see the temperature on mine, it's fine. Because, again, restriction enzymes are kind of wimpy. So if this is at 94 when you put it in because you're not paying attention, you might kill the restriction enzyme. And then I'm going to now go to the program and library. I have one restrict, and this is just going to be 37 degrees for 15 minutes. Oh, no power. Turned it off. There we go. That should be on now. It was a little hot. It's cooling down. It should be fine. All right, so it's restricting. Let's talk about the next steps. There we go. Oh, I forgot to say we put in the restriction enzyme there. Okay, so again, remind you, we're looking for this RFLP, and the FNU4H1 site. If it's the taster allele, it will cut it into two. If it's the non-taster allele, it will not cut it. So how do we see that? Well, we're going to run a gel. So an agarose electrophoresis gel. An agarose gel, here this is our casting trays. So this is what we actually make our gels here. I have one that I can show you. I'll just make this bigger so you can see it. So this is a gel. It looks like this. They're in trays. They have these wells at the top. You can see those are little like pockets that we're going to put our DNA in. And this gel is made from something called agarose, which is it's a seaweed extract. It's a carbohydrate, and it ends up being, I'm not going to use this one, but you can see it ends up being a lot the consistency of Jello, kind of like thick Jello, but it's made from a carbohydrate instead of being made from a protein like Jello is. So I'll go back here real quick. So here we go. So this is, if you were to zoom in on your gel, you would see that it has this structure. So this structure is like a complex web net that the DNA has to get pulled through. So you put your DNA up in the top. Oh, and I just lost 1 second. Here we go. You put your DNA up in the top in these wells like I showed you, and then an electric charge pulls your DNA through this complex web. And as it does that, it's as if you can imagine it like these two balls. So these two balls represent DNA of different sizes, and this is the net that it has to get pulled through. They both have a negative charge, and so they get pulled towards the positively charged DNA. We turn it on, it runs that far, and then we turn it back off again, and it stops. And so from there, we can tell relatively how big our different sizes of DNA are. So our smaller DNA moved farther than our larger DNA. That's what's going to happen in our gel, and we're going to go ahead and load it, and then we will share some results. And excuse me, I just got my gloves. I changed one glove because I handled the gel, and then here we go. Okay, so great. So we're going to I have 3 samples here to put in. I'm going to move back, switch my scene here, and I'm going to, so this is our electrophoresis system right here. This is our blue gel system. And the thing that you'll notice about it is that there's no external power supply, so it's all built into one. It doesn't look like a lot of traditional electrophoresis systems that you've probably used before. The power runs through the top, so there's electrodes going through the lid, and the gel that I'm going to use for this one is already in here. It's right there. So I'm going to go ahead and load it. For this, I believe it calls for 10 microliters of DNA ladder. So when you run a gel, the DNA will run in at different speeds based on how big it is. So smaller pieces will run faster and bigger pieces will go slower. But how far they move through the gel is just relative. So you need something to compare it to. That ladder that I just put in is DNA of known sizes, so I can compare my results to something. I'm now going to put in 12 microliters of my other samples. So these ones I have prepared. They're both of my DNA, and one I haven't restricted. Put this one in first. And just to load a gel, I like to come in and you just have these little wells there, but the DNA that we're putting in there has a dye in it, and that dye is heavier than the buffer, so it sinks down to the bottom. So you can just keep your your tip sort of above the well, and you just slowly release. I use my other hand to steady and all there. And then finally, we're going to use the one that I restricted, 12 microliters of that, so we can run them next to each other. And now I have other results for my whole family to show you, but I do just want to show you how quickly this can run, and that we will be able to see some results that I'll try to show you by the time we're done. We just pop the top on, and we hit the power button, and that light means that it's running. All right, so we're loaded, it's running. Here we go. So just quick little note. This is our casting system. The combs, they hide on the bottom. It's very easy to use. You can see just very small gels. One thing that we're releasing that we want people to know about are these gel agarose tabs. This has absolutely everything in it that you need to run to make a gel. So in one of these tabs, it has the agarose, the buffer, and the DNA stain to run one gel. So you just break open the tab, add water, microwave, and pour your gel. If you've ever, if you've used that blue stain and destain before, you shouldn't use that stuff just because it's such a pain in the neck. These results we'll be able to get in just one period. We use gel green as a stain, which is part of the newer generation of safe dyes to use. Okay, so let's look at what possible results could be. So again, we're going to put our DNA. We're going to put our DNA in the well up here, and a charge is going to pull it down. And in each one of these wells, remember, we're putting like billions of pieces of DNA. So it's billions of pieces being pulled through this, and how far they go depends on how big the DNA is. But they run all in the same size and shape, similar to what this is called a band. So it looks something like this. Now this is what our ladder will look like. We have DNA, it's 100, 200, 300, 400, and 500 base pairs, and then keep on going up, but we don't need to worry about those, so that we can tell how big our fragments are. So let's look at this. In lane A, we have 250, it is about 250 base pairs. That's how big our PCR product was. So this is supposed to be showing you what it could look like after you've added the restriction enzyme. This one is still the same size as the PCR product. B, okay, so this one has two fragments, one at 100 base pairs and one at 150 base pairs, and that adds up to a total of 250. This one has two fragments after we added our FNU4H1. And finally, we have an option. It could have three, right? So it could have the 250, and then it also has these two smaller ones. So you can think for a second what that may be. I'm going to tell you. But this A1, we put in the FNU4H1, nothing cut. That means that is, that's right. That means that this is in a different order over here. Sorry, I didn't mean to bring that up yet, but we'll leave it there for a second. This means that this did not cut with the FNU4H1. That means that all the DNA in that tube was non-taster DNA. In this one, all the DNA got cut in two, so all the DNA was taster DNA. In this one, the PCR made 250 base pair fragment off of one chromosome. All the fragments that got made could not be cut. Off the other chromosome, all the fragments that could be made were cut. So this would be a heterozygote, and in this case, heterozygotes can taste. So again, this is an actual result. It's kind of a faint gel, but just so you can see what they look like. This, again, going across, this would be a non-taster, a taster, and a heterozygote taster. So, the grand reveal, let's look at my family. So here's our ladder. So we have 100, 200, 300, 400, 500 base pairs. Okay, so the first thing I did was I ran all four PCR products before I'd added the restriction enzyme. So let's see what those look like. All right, so this one looks really nice right here. This one looks kind of messy. That happens sometimes. These ones look a little better. So this one didn't have a great DNA extraction. And we see something here, it's called primer dimer. Sometimes primers, if you see a band that's under 100 base pairs, it's because primers are like binding to themselves in weird ways, and they're making products that we're not interested in. Pretty much as a rule, if there's a band below 100 base pairs, right, if you didn't plan on it being there, it's some form of primer dimer and you just ignore it. So those primer dimers are more common when the DNA extraction isn't as good because it doesn't have the correct DNA to bind onto. Now I did this gel the other day. I said you need to use the DNA relatively quickly for some logistical reasons. These three, I needed an extra half an hour before I ran the gel, and so they were sitting around, or I'm sorry, before I set up the PCR, not ran the gel, before I set up the PCR. And so I think that's why these ended up with some primer dimer. I was going to rerun it to get a really pretty gel, but some of my colleagues said, like, no, show them that I can mess up a little bit too, but even if I do, we still get good results, okay? So let's look at who these people are and what it tells us. So dad and mom. I could taste it, and sure enough, I have off of one of my chromosomes, I made a 250 base pair fragment that got cut into two, so I have the 100 and 150. And then on my other chromosome, it was not cut. So I am a heterozygous taster. Mom, look at that. Even though it's not the best DNA extraction there, we see that we still get the same results. We have two smaller bands. We also have one bigger band. Mother is also a heterozygote. All right, for the kids, kid number one. Oh, I'm sorry, we're going to do a Punnett square first. Here we go. So Punnett square, what could mom and dad produce? What are the possibilities? Well, if you set up your Punnett square like this, right, we carry my t's down, mom carries her t's across, and we fill in the square, and you see that this pairing had the possibility to produce any type of child. So let's see what we get. Child number one, oh, we are 3 for 3 for heterozygotes, okay? Exact same thing we see here. Half the DNA was not cut, so off of one chromosome. We don't know if that chromosome came from mom or dad, but it came from one of us. It was not cut. The other chromosome, it was cut. And now, child number 2. All right, we are the most boring family in the world. We are a family of 4 heterozygotes. And sure enough, look at this. Either mom or dad gave her the big T on one chromosome, and either mom or dad gave her the little t. Oh, I just did that backwards. The big T would be down here. The taster got chopped in half. And on the other chromosome, she has the non-taster, which was not chopped in half. Okay, so again, if I had people outside my house to test, maybe I could have given you a more exciting gel, but just to show you these are authentic results, this is what we got. And we can say, well, was Mendel right? Did it work? Well, sure. We, for every one of the children that this pairing produced, there was a 50-50 shot. So 50-50 shot of getting heterozygote. We happen to do that twice. There they are. And we don't know, again, I don't know whether this daughter number 1 got the big T from me or mom, but I'm just filling it in here. And you can also say, all right, if this pairing were to make more children, they won't, but if they were to make more children, what would they be? And so the rules of Mendel say that it's just probability, so we're going to go out the same way. It's always going to be a 75% chance that the pairings from this mating would have tasting ability, and a 25% chance of non-tasting ability, and then a 25% chance of being big T, 25% chance of little t, and a 50% chance of being heterozygote. Okay, so just to wrap things up, and we do have some more after this, we're going to answer some questions and tell some extensions and things to do. But just this entire lab, hopefully you see how I took you, I sort of took you in reverse order, but we started out talking about a phenotype. We looked at all the different genotypes. We're able to connect it through the messenger RNA to talk about the different types of proteins that you could make and how those proteins interact in a signal transduction pathway to give you a differential response. One thing that we really want to just note here, remember genetics is really messy. Okay, so we use PTC because it is simple, because it's a really clear example, but even PTC doesn't really explain this everything, right? Remember that mom and daughter number one really, really, really hate the taste of PTC. Dad and daughter number two don't mind it that much. Like, don't like it, but don't mind it that much. That cannot be explained by genotype. So even this example is more underlying complexity than we talked about. And even this, one of the reasons we use it, again, it's simple. It's the only taste receptor that we know of that works in this very simple, you can taste it, non-taste it, dominant recessive way. So trying to suggest that this is how all taste works would be really problematic. And as I said before, there's actually 8 different PTC tasting alleles. Those different SNPs come in every combination, and so that's 8 different possibilities. Now the 2 that we talked about today cover over 93% of the variation. 2 more cover like another 6% of the variation, and those 2, it's just important to note, those 2, this test will work the same way. So those 2, if you're able to taste it, this test will come up positive in the same way. And I'll talk about that a little bit at the very, very end. Questions. Actually, before questions, I just want to see if I can do one thing. Yeah, I can. So I'm going to switch, and this might get a little difficult to see for 1 second, but I just want to show you the gel that I started running. You've seen how long it's been running. So we don't have super clear results, but yeah, we can start to see. And I just, again, just to show you how quickly you can get results here. Okay, we're coming in. It's doing this with a laptop. Usually you do this with a cell phone. Oh, and can I get there? Oh no, there they are. There they are, you can see them. Oh, this is really hard to do with a laptop. The idea, normally you do that with a cell phone, but it was a good effort. Normally you do that with a cell phone and you can see the picture very quickly. So I already have results that I can start to interpret there. And I don't know how long it's been since I've been running it, but just a few minutes. Sorry, that got kind of ugly there for a second. We're back. Okay, so just to wrap things up, folks submitted some questions. Great one. Companies like 23andMe give you your PTC genotype. Is this how they test it? No, they don't use gel electrophoresis. My understanding is companies like 23andMe use technology called SNP chips, where they have genotyped the human genome for like tens of thousands of these SNPs. PTC is one of them. They then use little probes and they wash your DNA over it. And if your DNA has a SNP that matches, it will bind. And they can visualize that in a machine. And so they use SNPs and they use binding of SNPs to see what your genotype is. So companies like that, they don't actually sequence your DNA. They just measure you at tens of thousands of different SNPs along your genome, and that gives them enough information that they can start to give you genotype information. How long does this lab take? So I told about throughout, but just a reminder, typically we say you can do it in 45 minutes and 45 minutes, 2 class periods. The DNA from a PCR is stable, so you can just set the PCR up and leave and it's fine. That second 45 minutes tends to be a little tighter, so if you do want to stretch things out, you can stop it after the restriction digest. You just want to make sure that you put your tubes in the freezer at that point. Restriction enzymes, sometimes after they've cut all their DNA, they get a little greedy and they start cutting DNA in random places. It's something called star activity, but if you freeze it, it should be good for a while. And then if you do it all at once, give yourself 2.5 hours and you should be able to do the whole lab. Does this relate to super tasting? So super tasting is something that people really like to talk about, probably because it It makes you feel like you have a superpower. PTC does not relate to super tasting. It is not the same thing. Super tasting is a fun thing to investigate. One of the things it's related to is how many of those fungiform papillae you have on your tongue. Something I used to do in my class as part of this, just to explore taste perception. You can dye your tongue blue with some blue food coloring. You just put a little blue food coloring on a Q-tip, and you rub your tongue, and then you cut out a 1 square centimeter out of a note card. You stick out your tongue and you take a picture of it, and you can count the differences in papillae, and that relates directly to super tasting. And it's fun to do. I don't really trust that students get the exact counts, but they get counts ranging between like 10 in a square centimeter to like over 100. So you can see that variation very clearly in a class. It's fun to do. Is there any way to replicate this without PTC paper? Again, unfortunately, we're all at home. I don't think there is a good way to do it. That's why we use PTC, because it's such a special chemical. PTC is chemically similar to some food molecules that are found in brassica plants like broccoli, but there is no other molecule that we know of that you just put on your tongue and you taste it or you don't taste it. So again, thank you for everyone who's been here. I want to answer some questions for teachers before we finally wrap it up. I just want to note that this is a great activity to do for all levels. Like introductory, you just want to match genotype to phenotype. You've done it, you're done. Kids see what they are, able to taste it, they predict what they are, they see it. It's a great lab to do that. But for advanced to AP levels or even intra-college levels, this is a great lab because you can connect those single nucleotide changes, you can line them up with the protein, you can see how those changes affect the protein and how that could possibly even relate to signal transduction. Again, Gprotein coupled receptors are a huge thing that AP students are always trying to cram for. This is a great example to have in their back pocket. As part of that, there's some really great extensions you can do with this lab. You can calculate Hardy-Weinberg equilibrium for your class. You can, a lot of times people are looking for real life examples of calculating Hardy-Weinberg. So this is a great one to do that. You can investigate population differences in relation to human evolution. There's a great article, which is one of the final slides here, which we'll just leave as part of the PowerPoint if you want to download it. That talks about that looks at PTC, or I'm sorry, TAS2R38 variants across the globe, and it follows a very classic example where there's more variation in sub-Saharan Africa and less as you leave, because again, sub-Saharan Africa is the cradle of human evolution, and so that's generally where you might find more human variation. That is just also a thing to note. If you teach with populations that have a lot of students who are of African descent, those rarer alleles that I talked about are a little more common. Again, the test still works, so you can still do this test, but it may be something that you want to bring up and talk about. Genetic drift or balancing selection. Why are there two alleles and one of them doesn't seem to work? How could that survive in a population? There's different arguments and people don't really know why. One could be just it doesn't matter. Maybe we don't really use PTC, the TAS2R taster gene that much at all, or the receptor that much, and so it doesn't matter if it's non-functional. There's some people that think that that second allele is actually tasting for something else, and we just don't know what it is. But again, we don't know. And just finally, miraculin. My colleague used to like to frame this lab this way. Miraculin is an extract from a berry you put on a tongue, and it just drastically changes your sense of taste. So where I framed it with like, is your blue my red, you can use miraculin and you can frame this whole thing as like, what do you know about taste? Take this and then taste something and it will taste different to you. We can change your perception. That's just another great way to sort of crack open these ideas of perception with students. We have a whole bunch of labs. Check them all out. As far as these webinars, we did another webinar on our BioBit Central Dogma Lab last week. You can find it on our YouTube channel. We're going to be doing a webinar next week on CRISPR. We don't currently have a lab for that, but we'll be talking about it with you. And then we have other webinars coming up on our quantitative PCR lab right here, and also our where our food safety lab. I know. Oh, I'm looking at the right thing. Our food safety lab as well coming up. So we have more webinars coming up. Stay tuned to our channel. Follow our emails or our Twitter feeds, and we'll let you know about them. That's what I got. Thank you so much. Thank you to everyone that is still here. We really appreciate you spending this time with us. We hope that this has been valuable to you. And again, to teachers out there, I cannot imagine being asked to teach online at a moment's notice. So we really respect the job that you are doing. To students who are still out there listening to me, I have 2 high school students who are, locked in their rooms all day on the computer. We realize that there's a difficult transition. So thank you to everybody for what you're doing, and hopefully that we can, just help make this a little easier for you going forward. Thanks a lot, and have a great day.